DNA. DNA stands for deoxyribonucleic acid. And it is a double helix structure, contains genetic material needs for transporting the genes character characters from one offspring to another. Also required to control different functions of cell. अच्छा, RNA, ribonucleic acid. A single strand a single strand structure if it is double strand, then it is found in viruses, it is usually meant for protein synthesis, synthesized from DNA of the cell. As far as the composition of DNA or RNA is concerned, they are made up of ribose sugar, phosphates, and nitrogenous bases. Same as with the composition of RNA, but there is a slight difference between them. Ribose sugar, phosphates, nitrogenous bases. As far as the ribose sugar of the DNA is concerned, it is deoxyribose sugar. That is, oxygen is removed from third carbon of the ribose or 5 carbon sugar. Okay. Acha, collectively, all of them made the nucleotide. It made the nucleotide. Acha. So there is a difference between the structure of the DNA and RNA, composition of DNA and RNA. Now let's have a look on the structure. As we know that it is made up of ribose sugar, that is 5 carbon sugar. It's like look like this. There's an oxygen and it combines with 5 carbons. One, two, three, four, and the fifth carbon is there on the top. Here, ribose sugar has got hydroxyl line. Here also OHM. This one is the first carbon, second, third, fourth, and fifth. In DNA, the oxygen is removed from the second carbon. Okay, then it will become the deoxyribose sugar. Okay, and on that, in between first carbon and second carbon, the nitrogenous bases are going to attach with it. N2 bases and nitrogenous bases. And what are the nitrogenous bases? I will tell you. And on the fifth carbon, 3 phosphate ions are attached. Okay, 3 phosphate ions are attached. Now, same as with the ribose nucleus, nucleic acid, ribose sugar, what happens that in ribose sugar, there is no loss of oxygen, but rather have 5 carbon sugar structure. same as that of DNA. Nitrogenous bases. Okay. As far as the nitrogenous bases attached in the RNA is concerned, this nitrogenous base is actually attached on the first carbon. ठीक है, ये फर्स्ट कार्बन पे अटैच होती है, जबकि ये वाली जो नाइट्रोजनस बेस होती है डीऑक्सीराइबोज की जो होती है फिर सेकंड कार्बन पे अटैच हुई होती है. As far as phosphate PO4 minus 3, PO4 minus 3 and PO4 minus 3 ions are concerned, in case of DNA it is attached on the fifth carbon, whereas phosphate PO4 minus 3 is also attached on the fifth carbon, same as that like the deoxyribonucleic acid in the ribonucleic acid. ठीक है, फर्स्ट कार्बन, सेकंड कार्बन, थर्ड कार्बन, फोर्थ कार्बन, फिफ्थ कार्बन पे यहाँ पे भी फॉस्फेट के तीन आंसर दे सकते हैं, PO4 minus 3, PO4 minus 3 and PO4 minus 3. Okay, this whole structure is also called the nucleotide. Nucleotide. This is DNA nucleotide and this is RNA nucleotide. Okay, shall I clean the board?
Yes, ma'am.
DNA sugar phosphate backbone formation. As we know that DNA is made up of nucleotides. Okay? So the nucleotide structure we had discussed, 5 carbon sugar, okay, and has got 3 phosphate molecules is there, and here the nitrogenous base. Okay. As far as this nitrogenous base is concerned, it is attached on the second carbon. Okay. And in RNA, it is attached on the first carbon. Nitrogenous bases are basically adenine, thymine, guanine, and cytosine. For DNA, okay. So they attach one by one with each and every nucleotide. This one is the oxygen. Okay. So how the nucleotides are attached? First of all, in a DNA, in nucleus, there are so many nucleotides are formed. 5 carbon sugar, 5 carbon sugar with 3 phosphate, and suppose here the adenine is attached. I am writing instead of adenine red color. Okay, and another 5 carbon sugar nucleotide is formed. Suppose for the guanine, so different types of nucleotides are formed in the uh, nucleus or nucleoplasm by the cell. ठीक है, adenine, guanine, and then एक साइटोसिन दोनों बना लेते हैं. And another one is the thiamine. They are lined up to make the sugar phosphate backbone. Okay, how they lined up? Actually, the first carbon and the first carbon, second carbon, third carbon, fourth carbon, and fifth carbon. So what happens? The fifth carbon is attached with the third carbon. ठीक है? The fifth carbon and the third carbon. They are going to attach parallel, attach side by side, or what can I say, one by one, to make the sugar phosphate backbone. So first I'm taking the adenine. Okay, here you go. Adenine ले लो. ठीक है, अब इसमें जो है फिफ्थ कार्बन के ऊपर 3 फॉस्फेट के मॉलिक्यूल्स हैं. Okay, this one is the first, second, third, fourth, and then fifth. अब होगा क्या कि इसमें से फॉस्फेट का मॉलिक्यूल एक निकल जाएगा और उसकी जगह पे फिफ्थ कार्बन आके लग जाएगा. Like this. ठीक है, अच्छा, अब यहाँ पे फर्स्ट करो गुआनिन आ गई न्यूक्लियोटाइड, जिसपे गुआनिन साथ लगी है नाइट्रोजनस बेस. समझ आ रहा है ना?
Yes.
और इसके साथ ये फिफ्थ कार्बन है, ये अटैच हो जाएगा. अब ये जो फिफ्थ कार्बन है और ये जो थर्ड कार्बन है, इसके दरमियान बॉन्ड बनेगा, दैट इज कॉल्ड फॉस्फोडाइस्टर बॉन्ड. ठीक है, ठीक है, ये फिफ्थ कार्बन इसका और इसका थर्ड कार्बन ये वाला। अच्छा, इसके नीचे फिर एक और आगे लगेगा, साइड बाय साइड इस तरह ये लगते चले जाएंगे। ये देखो, ठीक है। अब इसके और इसके बीच में, फिफ्थ कार्बन के और थर्ड कार्बन के बीच में फिर बॉन्ड फॉर्म होगा, दैट इज कॉल्ड फॉस्फोडाइस्टर बॉन्ड। ये देखो, फिफ्थ कार्बन और ये थर्ड कार्बन। फॉस्फोडाइस्टर बॉन्ड क्यों है? दो फॉस्फेट के मॉलिक्यूल है, दैट्स व्हाई डाइस्टर बॉन्ड कहलाएगा। एक फॉस्फेट निकल के जिस वजह से 3 और 5 के दरमियान बॉन्ड बन गया। यहाँ पर मैं लगा देती हूँ, चलो थायमिन। Thiamine. And that's how the sugar phosphate backbone is formed. This one is the phosphodiesterone. Understand? Phosphodiesterone. अच्छा, ये तो एक शुगर फॉस्फेट बैकबोन बनी। ये क्या है? ओवरऑल शुगर फॉस्फेट बैकबोन सिंगल स्ट्रैंड की। सिंगल स्ट्रैंड ऑफ डीएनए। लेकिन डीएनए सिंगल स्ट्रैंड नहीं होता है, डबल स्ट्रैंड होता है, डबल हेलिक्स होता है, डबल स्ट्रैंड होता है। So another strand should be in front of that, this strand. How this form? Let's see. Same with these nucleotides. ये कुछ इस तरह बनेंगे कि ये उल्टे बनेंगे। That means कि the structure is the same, but the third carbon is on the top. As we know that adenine pair with the thiamine वाला जो न्यूक्लियोटाइड है वो पूरा इसके सामने आ जाएगा। I'm making the nucleotide first in order to line up the sugar phosphate backbone, वरना वो फिर ऊंची नीची बनती है। For the drawing purpose, I am doing this. Now here it is formed in an inverted way. Okay, this one is the first, second, third, and fourth. Okay, so here it is the first, second, and third carbon, and this one is the fourth one or fifth one carbon is here. Now third is on the top, fifth is on the downside. That is tail is on the, or what can I say, foot is on the top and head is on the downside. Okay. This one is the third carbon. This one is the second. This one is the first. This one is the fourth, and then fifth. between third and fifth, the bond is formed, phosphodiester bond. Okay, same is with this case. And between fifth and third or third and fifth carbon, phosphodiester bond is formed. Acha, this is how the second strand is formed, that is completed. In between these two strands, here it is the base pairs, that is nitrogen base pair, adenine pair with thymine, guanine pair with cytosine, and then adenine. Okay. As far as the bonding is concerned, in between adenine and thymine, double hydrogen bonding is formed, double hydrogen bond, and in between guanine and cytosine, triple hydrogen bond is formed. Adenine plus thymine, double hydrogen bonding, or guanine and cytosine, triple hydrogen bonding is there. Okay, यहाँ तक बात समझ में आई? As far as double hydrogen bonding is a weak bonding, and triple hydrogen bonding is the strong bonding as compared to double bond. Whereas phosphodiester bond is the strongest bond. Okay, that's how the two helix of the DNA is formed. Okay, this one is the double helix structure. Double sugar phosphate backbone, not helix. अभी helix की form में आया नहीं. Phosphate backbone, VB. Now, when we look at this structure of the DNA, we see that there is a double helix structure, right?
Yes.
Like this. And in between that double helix structure, nitrogenous bases are attached and bonded. Like this. But as we go in a deep, when we talk about the replication of DNA or the formation of the DNA double helix structure, we find it has the two groups. These are the minor groups and these are the major groups. This one is the major groove. Okay. Minor grooves are more twisted and major grooves are less twisted. Why? because wherever the tight bonding of guanine and cytosine has come up, or the ratio of guanine and cytosine bonding is high, then the twist becomes the minor groove one. Okay, ज़्यादा वो turn हो जाता है because of tight, strong bonding of guanine and cytosine. That is triple bond. Bonds. Whereas as far as the major grooves are concerned, here light or weak hydrogen bonding ratio is high. Is high. That is adenine and thymine bonded. So the grooves become loosely packed, or what can I say, somehow less tightly packed. And when we look at that twisting, the third carbon facing the fifth carbon of ribose sugar is the fifth and this is the third. Okay, that's how the sugar phosphate backbone is formed, and along with that, the DNA double helix structure is formed. Okay, now the replication. Should I clean the board?
1 minute.
Okay. Okay, now we will study the replication of the DNA or RNA. In both replication of DNA and RNA, the following enzymes are involved. For unwinding the DNA, a double helix structure, helicase enzyme is used. Enzyme is used for unwinding of DNA, and polymerase is used to make sugar phosphate new backbone, as well as winding of DNA. As far as unwinding of DNA is concerned, it is also done by the temperature fluctuation. Unwinding of DNA. Two methods, in fact, three methods. Number one, high temperature. Number two, change in pH. Number three, helicase enzyme. High temperature and change in pH. Outside cell, outside लिविंग बॉडी, ये दो मेथड यूज किए जाते हैं for unwinding the DNA, whereas in the inside helicase enzyme. Inside living body. अगर रेप्लिकेशन करवानी है तो ये मेथड होगा, हेलिकेज एंजाइम आएगा। ठीक है, as far as high temperature और change in pH is concerned, जब तक टेम्परेचर हाई रहेगा तब तक ये अनवाइंड फॉर्म होगा, जैसे टेम्परेचर नॉर्मल पे आएगा ये दोबारा बाइंड हो जाएगा। सेम यही है, पीएच अगर उसका डिजायर्ड लेवल के ऊपर आ जाएगा तो दोबारा से बाइंड हो जाएगा। अच्छा, as far as unwinding is concerned, unwinding of DNA is concerned, it takes place from the major groups. Usually कहीं से भी हो सकती है। Suppose helicase comes and acts on that group. तो ये यहाँ पे जो है ना हाइड्रोजन बॉन्ड जो होगा उसको तोड़ना शुरू करेगा। तो हाउ इट लुक्स लाइक, इट गेट्स अनवाइंड और सीजर शेप जैसे होता है ना इस तरह बन जाएगा और bases gets apart, nitrogenous bases. The bond between them gets apart. Hydrogen bonded to duality. Now what happens? Polymerase sorry. Polymerase start acting on that area and brings nucleotides. This nucleotides, okay, and starts making another strand. Now, one thing you should notice that 3 strand and 5 strand, 5 strand and 3 strand शुगर फॉस्फेट बॉन्डिंग का। ठीक है, अब अगर फर्स्ट करो कि ये जो है ना ये वाला कौन सा वाला स्ट्रैंड है, इट्स 3 स्ट्रैंड, तो नीचे वाला 5 है, तो
yes,
जो 3 स्ट्रैंड है इधर की तरफ से 5 बनना शुरू होगा और 5 की तरफ जाता हुआ जो होगा वो 3 होगा। दैट मीन्स के हियर ये 5 होगा और ये क्या होगा 3 होगा।
3,
ठीक है, और सेम इज केस विद दैट, ये वाला जो स्ट्रैंड इधर बनना शुरू होगा ये 3 होगा और ये वाला 5 होगा।
5,
Understand? So that's how
yes,
the replication takes place. If the double helix is being formed on the both side, the strands are going to be formed. If the messenger RNA is going to be formed, then what will happen? Only one strand is formed. This looks like ये इस तरह अलग अलग हो जाएंगे और यहाँ बीच में ना मैसेंजर RNA बन जाएगा 5 and 3 स्ट्रैंड का। ठीक है, और इसके ऊपर न्यूक्लियोटाइड जो होंगे वो आगे ऑपोजिट के लगने शुरू हो जाएंगे। When it gets completed, it gets out in between that loop having the nucleotides on it. And it gets again unwound. Same is case with the transfer RNA. इसी तरीके से transfer RNA बन जाएगा। When it gets out, it gets folded. Okay, एक इसके ऊपर जो है अमीनो एसिड कोई लगा हुआ होगा, सिंगल अमीनो एसिड लगा हुआ होगा, ठीक है। और यहाँ पे सॉरी 3 कोडॉन एंटीकोडॉन जो होंगे वो भी लगे होंगे। दैट मीन्स 3 न्यूक्लियोटाइड यहाँ पे फोल्ड हो जाएंगे। वैसे तो न्यूक्लियोटाइड पूरे में लगे हुए होते हैं, लेकिन these 3 specific nucleotides are said to be the anticodon. न्यूक्लियोटाइड सब जगह लगे हुए होते हैं, लेकिन ये वाले जो होंगे these are the anticodons. एंटीकोडॉन्स, और ये होंगे बेसिकली न्यूक्लियोटाइड, ठीक है। अच्छा, ये जो है ये कहलाते हैं कोडॉन्स, मैसेंजर आरएनए के जो होते हैं। ये भी सब किससे मिलके बने हुए होते हैं, न्यूक्लियोटाइड से मिलके बने हुए। अंडरस्टैंड?
Yes.
ठीक है।